503 in the house already. Joe's. And valid point 503. Some people are probably going to forget. But hopefully the notifications go out. Now that I am back to Wednesdays. And should be back to Wednesdays for some time. Let's see. Try to make that a little bit more clear there. Yep, I can see that the notification's going out because I just got one. So, oh, here's Olivia. Excellent. Exotics galore. Excellent. Had a pretty good set of questions in Patreon. Looking forward to talking about some of those topics. I see Shane's Vlogs is here. Wendy Hickson is here. Great. Young lad. Starting to build up. 15 people here already. Let's hit that like button and get some algorithm love. Happy back to school. Yes, indeed. Many of us are back to that aspect of life, and summer is, in a sense, over. But here it's actually very hot. It's been 102 degrees today, which is hot for September. It gets warmer than that, obviously. Earlier in the year, in July and August, it can get warmer than that. But 102 in September is a little excessive for where I am now. It's kind of unusual. KK Pania, I'm not sure how to say your name, but welcome. I think I welcomed young lad already. Excellent. Well, 17 people. Should we start hitting some questions? I think we should. And patrons will hopefully appreciate that we're hitting the questions early. It's easier to catch them when they know that they are at the beginning of the live stream. So we've got 5 comments here and our questions. Okay, one is Sandy Sizemore is asking about the morning gecko enclosure. Says there's mold in substrate and leaf litter. How much is okay when to toss it? So let's see. Well, if I see a little bit of mold in a new vivarium, I'm not going to worry about it too much. But if I see it if I see a lot of very fluffy stuff, I know it's going to be bad for the isopods and possibly for other small creatures. Also, if it's like a black mold or something, I would start to worry about that too, obviously. But a mold bloom in the early stages of an enclosure, bioactive vivarium, not unusual. And that's why it's ideal to have the springtails and isopods in there, of course, first for a month or two, kind of let that die down before you put anything else in there. And maybe that's what you're doing, or maybe you're talking about something completely different from that. But I would generally say if it's in those early stages and there's enough ventilation, I'd probably kind of watch it and see if it gets so it's covering much of the substrate. I'm going to take some of it out for sure, and probably take out geckos and any macrofauna and just kind of focus on getting the springtails going and getting that under control. But if I see a little bit, I'm not going to worry. If I see it spreading steadily and not showing any signs of slowing down, I'll probably address it. Also wanted to ask, Sandy wanted to ask, what is your best-selling isopod? Numbers, not money. Okay. If we're talking species, it could be Armadillidium vulgare because there are so many morphs and they're popular. Porcelionides pruinosus sells a lot. Armadillidium maculatum, the zebra isopod, sells a lot. Would be hard to say. I would have to go back and look at my books and kind of compile that over the past couple of years. See what my records say about which ones I sell the most. But those are some that come to my mind. Decent number of Porceliolavis too, but I think I sell more Armadillidium vulgare and Porcelionides pruinosus than that. Just 1 second. I need to take a drink here. My throat is, sorry, just a bit scratchy today. So, thank you. Like the shirt, still available on Spring. What used to be Teespring is now Spring at the Aquarimax store. Merch shelf is connected to the YouTube channel, so it's easy to find. This shirt may not be available a whole lot longer, but it's available now. And let's see, invertebrate dudes in the house. Excellent. Nice to see you here. So Kekepania wants to know, what can I do about mites? I was told they were soil mites. They seem to be killing off my springtails and getting in all my containers. They are orange and look like a tick. Okay. Oh, and Sahar's here. Excellent. Hi, Sahar. So about the mites, if they are soil mites, and they may be predatory, which is not to say parasitic, so not the same kind of mites that could be dangerous to reptiles. Predatory meaning they eat things like springtails and like other mites and so on. If that's what you've got, because if they're killing off the springtails, actually hunting them down, then you do have predatory mites. If you feel like it's more like they're out competing the springtails and that's something else, those just might be grain mites. But springtails themselves will often help outcompete grain mites, so these may be predatory mites. If they're killing off the springtails, one of the easiest things to do with the predatory mites, because they're going to eat soil mites, so you don't need to worry about those. I mean, they're going to eat grain mites, so you don't need to worry about those while they're there. Kind of let them run their course. Once they run out of food, they will tend to die off, and then you can reintroduce springtails, and then you don't have much of a problem because the predatory mites will be gone. I have done that with containers before, so that could be what you have, but it can be difficult to confirm what type of mite you have without, some close scrutiny. Let's put it that way. Savannah, welcome. Excellent. Going to be putting in another order. That's awesome. Thank you. Thank you for showing up here today, and I'm glad you've appreciated the orders that you've done. Okay. So yeah, Invertebrate Dude is saying something similar along the lines of what I was saying, I guess. Specialized in feeding on springtails, fungus gnats, and other mites. So that's the bright side, is that they will get rid of the mites and the fungus gnats. So good point, Invertebrate Dude. Warren is here. Welcome. Mr. and Mrs. Morelia here as well. Excellent. I'm getting close to the point, Mr. and Mrs. Morelia, where I have orange creams for you. Really close. So just updating you there. And Stephen Wright, greetings and welcome. Good to see you here as well. The Keep, Theropod Hunter. I know I'm going to be missing people. I'm doing what I can. But Victor Olson here is here as well. Sorry, I'm getting confused. Okay, I got another question from Patreon. Abby Manali says, what do you know about Cubaris species blonde ducky? I've noticed a fully yellow adult in my rubber ducky colony. Excellent. I would like to isolate it in similar individuals and hopefully get them to reproduce. I've also noticed that young rubber duckies are fully yellow. At what point do they develop their darker colors? Do you know if the blonde color is a recessive trait? I've been looking for information on the blonde duckies, but cannot seem to find anything solid regarding genetics. So, great question. One thing about the rubber duckies, and just about Cubaris in general, which many of you know, is that the taxonomy for the Cubaris genus is a train wreck, in that there are plenty of species that are just dumped into the Cubaris genus without really belonging there. It's sort of a catch-all category. And that the common names that are given to them, or the pseudo-scientific names, maybe we can call it that, because people will say Cubaris species blah blah blah, and use a random title for them. It's not really a scientific name. It's just sort of a placeholder name, and it's not going to be very reliable. And we have one issue in that there is a species that is sometimes referred to as Cubaris species blonde ducky, and then there's the color morph of rubber duckies that is also referred to as blonde ducky, so that can lead to some confusion. But from what I understand about the blonde duckies that are descendants of just normal colored rubber duckies and are therefore just a color morph of them, is that, well, I think it was Dan Vivas, from whom I purchased my first rubber duckies, who said that you need to select for the dark body on the rubber duckies in order to keep the trait. If you don't, the colors will just kind of pale over the generations. I have seen some of those paler ones in my colony. I started out with some really solid dark bodies on mine, and now I'm seeing some more of the paler individuals, and so I need to actually go through and separate more of those out more carefully. But I can't say exactly what kind of genetics are going on there. With very small populations and very small founding populations, it's hard to say if we're dealing with genetics, or if we're dealing with just simple recessive trait or whatever we're dealing with, that kind of stuff. So I can't tell you if they're recessive or not, but I will say that it has been stated that the blondes will eventually kind of dilute the colony, so you will end up with mostly blondes, which kind of sounds like it's not a recessive trait, but it's hard to say. Also, I've heard people say, well, don't pay more for blonde duckies because they're just culls from a normal rubber ducky bin, but it seems like that ship has kind of sailed in that the blonde duckies become popular, and they look cool too. Now, the fully yellow adult that Abby was talking about, that sounds actually pretty fantastic, meaning just as yellow as the head. That sounds awesome, because that looks like the plastic rubber ducky if you have the orange beak and then just the full yellow body and the little black eyes. How cool would that be? So if that's what you're talking about, that's fantastic, and I hope it works for you. Let's see. So that is about the extent of my knowledge on that. If you have specific follow-up questions, I'll do my best, but that's all I can think about. That's all I know about that. Another drink, sorry. I don't know what my throat's doing today. So Sandy also has a question about fruit flies. Wanted to talk about hobbyist fruit flies. How and why do they start flying? Sure surprised me the other day when my Heidii went up, up, and away. Why are the Melanogaster and Heidii the most popular for the hobby? What about Bazati, golden Heidii, and Turkish flyers? Okay, so let's talk first about the three species that are available in the hobby. We've got Heidii, or Heidei. They are the largest fruit fly in common culture. In culture, as far as I know at all, with the exception of possibly species that are being studied in labs and not released in the hobby. But they are the larger ones. The mutation that is in the hobby is one that causes issues with the muscles in the wings, or something like that. The wings themselves, of course, develop, but there's an issue with the muscles in the wings, and they can't do their thing, or something like that. Or I don't know if it's the muscles or the nerves or something. At any rate, the flies are unable to fly, but they are fully winged. Then we have Melanogaster, which is the smaller one, and that comes in a few different varieties. It comes in the apteris, which means wingless type. I have that type. It's my favorite to deal with. And then there are some winged ones. I believe the Turkish flyers or the Turkish gliders are another mutation of the Melanogaster, and they do have wings, and the wings are semi-functional. In other words, they can go for short distances. They can't really fly around. They're not going to fly around a room, but they can jump off of something and glide. They can sort of hop and go a little distance, something like that. I kept some Melanogaster that answered that description. Not sure if they were Turkish gliders or not, but I didn't really like keeping them. It was kind of, someone dropped some off for me when we were doing a bug trade or something like that, and they said, here, try these fruit flies. I've been working with these. And the advantage to them, whether or not they were the Turkish gliders, they were ones that could fly about 1 inch and sort of looped around. The advantage to them is that they move very erratically and very fast, and they're very, very, very attractive to a lot of small predators like dart frogs. They're amazing. They give the frogs a little bit of more workout. They get to chase them, and it's a little bit more difficult than just lapping up these lazier, slower moving fruit flies. They also breed faster, from what I understand and from what I remember. The disadvantages are that they can escape a little bit more easily because they can actually perform these longer hops, almost kind of abortive flight attempts that can get them, an inch or two, and that can be a little more difficult to deal with when you're dealing with flies, cultures, moving things around. And they also buzz audibly. When you have a group of them together, you can hear them buzzing. I was not a fan of that. It just kind of, I don't know, bothered me a little bit, and I think it bothered my wife more than it bothered me, and so I wasn't really a fan, so I stopped culturing those. And golden Heidii are just Heidii, but a different variety. Just a color morph, essentially, of those. And then Bazzotti, I have never worked with, but from my understand, Bazzotti is a different species. It's a true species, not just a morph from one of the other two, that is intermediate in size between the other two flies. So that can be useful when you need size gradations that are gradual between the two, because some species that you're feeding, like dart frogs, you can feed them melanogaster until they're big enough for high-DI. Not a problem. But if you wanted a gradation in there, you could feed them Bazzotti in the middle. I've also heard, from what I understand, I think it's Kyle at Roach Crossing mentioned on his website that Bazzotti tend to be good long-term producers. Like they'll start producing and they'll produce kind of smoothly in terms of numbers that they're producing out of a culture smoothly for a fairly long time. Melanogaster will do kind of like that too. High-DI tend to have a boom and a bust cycle. Like they take longer to produce. Once they produce, boom, they reproduce crazily, and then they kind of don't produce as much. And you can mitigate that to some extent by regular harvesting and not harvesting immediately. You let the first adult flies kind of mature for a few days and start reproducing, and then start harvesting. That helps. And then harvesting gradually over time helps. But they tend to do more of the boom and bust. And apparently the Bazzotti don't do that so much. So that's cool. And I think that hopefully that helps. I'm trying to think if there's anything else I had to say about that. Oh, okay. So I talked about the reproductive cycle and in a culture producing. Yeah, I think that's about all I had to say on the fruit flies, but I'm happy to answer any other questions if you happen to have them. Sandy, I think it's a great question. See, the beetle guy got here, and Luke, I am your father. So Luke, wondering about the dubia roaches breeding better in darker cages, so maybe rubber ducky would do better in a darker container? Don't know. Yeah, it's worth some experimentation for sure. Terminal Overkill has a question. So Terminal Overkill, you have a question about the temperature in your millipede enclosure. Heat pad is not helping. Interesting. How warm are you trying to get it? Oh, there's a super chat from Mr. and Mrs. Morelia. Okay, so Mr. and Mrs. Morelia, are there orange cream and a creamsicle morphs of Isopod? Talked to someone about the pruinosis orange cream and they said they are creamsicle. I've heard it both ways. From what I understand, they are merely different names. Of course, there are different origins out there for orange pied pruinosis. There are some that were produced and isolated the morph and produced the morph. I know Kyle Candelian did it. I've done it, and others have done it, as far as I know. So it's not difficult to do. It just takes some time. And I think different people name it different things. And probably it doesn't even necessarily mean it was a different producer. Someone could buy some as orange cream and then decide to sell it as creamsicle. So I've heard it both ways, and I think I've heard other terms for it as well. Can't think of any of the other terms right now, but yeah. As far as I know, they're the same thing, basically. But that is a great question and a great super chat, so thank you. See, Michael Hare is here as well. Great, welcome. The Keep says, my D. tenebrosus female is only eating minnows now. Is thiamine an issue with inverts? Good question. I'm not sure if, I mean, thiamine itself is the vitamin. Thiaminase is the enzyme that destroys the vitamin. So I can tell you that. So thiaminase is what causes the problem with garter snakes and other creatures that eat a diet. It can be a problem with fish. It can be a problem with reptiles. Other creatures, if they get thiaminase too regularly, it destroys all the thiamine in their diet, and they get a vitamin B12 deficiency, which leads to neurological damage. So, not sure how much it affects invertebrates. Good question. So, Terminal Overkill, what kind of millipedes are you getting? Oh, people are asking. Okay. Chintrobulus splendidus and a purple ivory. I think the ivory is not going to mind if it gets down into the 60s. I've never kept Centrobulus splendidus, so I'm not sure, but 80, and I know they're an African species, and I know Kyle at Roach Crossing keeps that species. I wonder if he would have some advice there. 60 does seem a little low for a tropical millipede. Maybe not excessively low, just because they spend so much time underground, and you go underground and you're getting into the 60s Fahrenheit. So, and Mr. and Mrs. Murley, that's a good point. Even talking about the same species, there are times when you get one morph title applied to one species and then it gets applied to another, and it does cause confusion that way too. So very valid concern, for sure. Definitely not something that we can take for granted that everybody uses the right terms, because they're not necessarily even the right terms to use in many cases besides the scientific names, and when it comes to morphs, it gets muddy. There's Hobby Strange Pets. Welcome. So, Sarah Lacrone is wondering if I rinse off eggshells before I give them to your isopods. I do not do that, but I do make sure that the membrane, and not the membrane, but the albumin that is attached to the yolk is not moist. It is no longer wet when I give it to them, because that can capture isopods. It's a very viscous substance, of course, and so I would not take a fresh eggshell and give it to them. What I usually do is put it in the oven and cook it. I've thought about putting it into a fruit dryer at a lower temperature so that you can potentially preserve some enzymes and whatnot in that connective egg membrane, and presumably in the albumin too, because when you're cooking it, of course, you're denaturing whatever protein enzymes are in there, all that stuff. But it's definitely better than them getting stuck on it, especially baby isopods are particularly vulnerable to getting stuck on it. So I always have cooked it, and then I grind them. I don't necessarily grind them super fine, but I grind them up, and that is what I do. So they never get that stuff wet. Sarah also had another question. Says, been really curious what it is about garter snakes that you like. What draws you to them? Sorry if you've already talked about this. I have them where I live, so it's odd to think of them as pets. They used to breed in the backyard of my parents' house every year until this one. The heat might have caused them to need to find a new home because everything was super dry. I feel you. That's how it is here too. Used to have a wild albino for a couple of years. He was a 2020 hatchling. I always thought of taking him in to possibly give him a longer life, but just didn't know if I should take him from the wild. Do you have any experience with taking in wild garters? Well, when I was a kid, I collected wild garters on occasion and tried to keep them as pets. Generally, I did not have escape-proof enclosures, nor much knowledge of how garter snakes were to be kept, so they generally escaped. But I have not kept wild garters long-term in my adult life at all. But I think taking an albino would make sense because it's not likely to survive an extremely long time. Probably not live out its even natural lifespan in the wild with an albino, so I could certainly see a rationale for doing that. So what draws me to garters? Well, let me tell you, I can collect garters here too, or could collect garters here too. There's a garter that lives on my back porch that I see occasionally. There is my neighbor next door has lots of garters in his yard, and I can go to any nearby body of water, a canyon, whatever, and I can see garter snakes. We have the wandering garter snake here, Thamnophis elegans vagans, I think it is, and we have valley garter snakes, which I think is Thamnophis fitchi, and then there's another one down in the southeast part of the state, and I don't get down there often enough. I can't remember which species we have down there, but there's another species down there. Anyway, so I can get garters. I mean, I can see garters around here, and I don't collect them to keep. I sometimes will pick them up and look at them, and whatever, short term, but I don't keep them. But there are a lot of things that attract me to garters, even though the garters seem maybe commonplace to a lot of people, and they are common in many parts of the U.S. and into Canada and into Mexico, and these areas. Part of it is that they're communal. The fact that you can keep different snakes together, even different species of garters together, you have to be careful about that depending on the situation. And certain species are more prone to cannibalism than others. Apparently the wandering garters can tend to be more cannibalistic than others. And at any rate, you have to be careful when you're feeding them, because one can accidentally swallow another one if they both start on the same food item. So there's that. But otherwise, they're communal. and you can keep more than one species at a time together, which I think is fantastic. So right now I have a fraternity vivarium with 3 males together, 2 different species, and I have a sorority which currently has 2 females in it, 2 different species, and then I have my baby enclosure with 4, all of the same species. My melanistic garters are in there. I love the fact that you can keep them together. They are social snakes. They've done studies on them and have determined that snakes form bonds with other snakes. Garter snakes specifically form bonds with other garters, and even after long periods of being separated from their buddies, they will find each other, and if they have made other buddies in the meantime, they will often integrate into one whole big buddy group, which I think is fantastic. And their social behaviors are very complex. I mean, you're not getting a lot of social behavior from a snake that is by itself, because it's not, by definition, obviously, because it's not interacting with other snakes. So the social behavior that they interact in there's a wide variety of interesting behaviors. I think it's enriching for them to interact with each other. It's fun to watch. Another one, they are diurnal. A lot of snakes that we keep in the hobby are not. So they might be more crepuscular or just nocturnal. And so you tend to see less of them, and that doesn't mean you'll never see them out in the daytime. But garter snakes you're going to see out a lot in the daytime active. That is when they are active. That's when they are going to be naturally active, so you don't have to try to shift their schedule or anything. And they're also extremely curious and interested in what you're doing. Your garters see you from across the room. In many cases, they will react to your presence from across the room. And then when I open the enclosure, I love this. I love the fact that when they see me and I open the enclosure, they will crawl up onto my arms and everything, just of their own accord, even if they're not hungry. And I've tested that. I have times where I'll offer the snakes food and they do not want it. They want nothing to do with the food, but they still want to climb up onto me and say hi. Almost like emerald tree skinks. They're not just going after food. They want to come jump on you. And garter snakes are very curious and seem to want to interact as well. And that is fantastic. Another thing about garter snakes is that, and this was one of my initial attractions, ones that I don't worry about as much now, but is that they don't necessarily need to be fed rodents. Now I do feed mine rodents because it's easy, it's inexpensive. In addition to other foods, I don't give them exclusively rodents, but there are some people for whom feeding rodents to snakes is a deal breaker. They're like, if I have to feed them rodents, I'm not going to have snakes. I would keep snakes otherwise, but I don't want to feed rodents. Well, with garter snakes, you have alternatives because you can feed them things like thiominase-free fish, like silver sides, that you can buy frozen at big box pet store. You have to make sure you're getting the right fish. But you can get the frozen silver sides, thaw those. Most of my garters like those. And so that's a vertebrate whole food prey item you can offer. You can offer night crawlers. Once again, I can go to the big box store and pick up a container of night crawlers, keep it in my bug fridge, and slowly just feed it off. And it's super easy to do. And the snakes really love earthworms. So you have options. Reptilinks is another food that I fed my garters, and they are rodent-free reptilinks. They have lagomorphs, they have rabbits in them, and they have quail. The mixture I've used is the rabbit quail mixture. And the garters will take the microlinks even when they're pretty small, and you can cut them up. Even if they're too small to handle the microlinks, you can cut them up with no problem. The bigger reptilinks too, you can cut those open and give the insides to the snakes, and they'll eat that. And they are fine on that. That's a good diet for them, and it's a whole diet because it's basically a whole rabbit and a whole quail mixed up. So you're getting the nutrition you would get from feeding a whole animal. I think they remove very small amounts of gut contents, but they leave some of the gut contents in. But the fur's going in, the bones are going in, all the organs are going in. So it is whole animal prey, essentially, but it is in a more palatable form for humans to see, if that makes sense. And my garters really like the reptilinks. So that's another thing. And then another is the amazing variety and beauty in garters. There are so many different species. You've got the dwarf garter snakes that stay really small, like Thamnophis butleri, I think it is. Is that the one I'm thinking of? A pretty small one. There's the giant garter snake, which reaches about 6 feet long. And there's the Lake Chapala garter snake, which is pretty comparable in size to the giant garter snake. It's pretty big. But then the colors and patterns. I mean, the The red side, the California red-sided garter snake, absolutely stunning. The melanistics are absolutely stunning. There's albinos, there are axanthics, there are high reds of different types. Absolutely fantastically beautiful. I love that. And I love the fact that you can play around with morphs. For example, I have melanistics right now, melanistic eastern garter snakes. I could get some flame eastern garter snakes and I could produce double hets. And then a generation later, I could be producing flames, normals, and melanistics all in the same litter. So pretty fun stuff. So those are some of the reasons why I love garter snakes, and I hope that helps. I just went off on garter snakes for a really long time, so I hope that's okay. All right, I'm trying to catch up on the chat. And Savannah, I do have a couple of videos on springtail culture care. If you just do a Aquarimax Springtail search on YouTube, totally find that. The most recent one I think is Springtail's The Easy Way or something like that. Is that what it's called? But there are a couple out there. One is a classic old one, but it still makes sense. It still works. Okay, so Mr. Zissou, hoping it gets down to the mid-80s for the daytime high for shipping by the end of September. Actually, next week is supposed to get down that low here. I don't know about wherever else, but like I said, it's 102 here, or was a little while ago, 102 today. So I don't know what temperature it is right now. But I'm not shipping any live things this week at all. I shipped some dry goods, but that's it. Let's see, do I have Zinger Gestroy for sale? Right now, my colony of Zingers is pretty small. Excuse me while I take my shoes off there. You think, well, one thing, I don't have a permit to sell Gestroy out of state right now, which is regrettable. My colony is still young. It's producing a lot of babies, but it's still a young colony. But that's the main thing, is that I just don't have the I don't have the permit at this time. I hope we can get permits for Gestroy going, because they're seriously my favorite armadillidium, and they really deserve to be spread around. Okay. And theropod hunter, that's an interesting question. Sometimes I do wonder if my garter snakes view me as part of their group. Part of the reason for that is something my wife has observed, because I try to socialize them with other people, and they're good with other people too. I bring them to animal outreach demonstrations and things like that, and they do really well. Not my baby melanistics, but you know, my other garters, my red-sided specifically, I've brought. They do really well. And I also will pass the snakes around and have different people in the family hold the snakes and so on, so they get used to different people. But my wife has noticed they seem to kind of, when they're with someone else, sometimes they'll zero in on me. They'll like look at me and like they're trying to get back to me, which is interesting. So maybe they do. Matt Bartley, how are your crested geckos doing? Pretty well. Got that new parthenogenic baby, which is interesting. Right now we have our original female and two of her offspring, I think is what we've got. One, the original one, Captain Squinty, and then we've got the little baby one that's just born, which people are just calling Little Dude. And theropod hunter, hey, I have kept a tiger salamander before. It's been a while, but I did have one as a pet, pretty easy one. Oh, cool, Mark, that's awesome. Glad to hear I helped get you into the hobby. So you're actually doing the wholesale breeding to a pet store. That's fantastic. It was not even possible to do that a few years ago. Selling isopods to a pet store wholesale would not even be a thing. So I think that's awesome. So just bugs. Yeah, it's not 120 here. So we're getting less of a heat wave than California is, but we generally have, you know, cooler summer temperatures than a lot of places in California. I think the warmest I've seen it in my state that I have experienced personally was 109. So still warm, but not as warm as 120. Wow. And that's a good point, Mantis God. You can put the leaves in the trash bags and bake them that way. I need to talk to my daughter about that. You could just go put a trash bag out in the sun and let it do its thing if it's this hot tomorrow. And random tea, yeah, I think snakes are underrated mentally. And if those of you who haven't seen Lori Torini's channel, or Reptiles in Research, should check it out. They have some cool things going on. Snake cognition is probably, they have a lot more potential than we give them credit for. And just bugs, that's another thing. 102 with very, very low humidity here is not as bad as high temperature and high humidity, for sure. Can't deny that. So Reuben, large enclosure, we bred together powder oranges and Oreo crumbles. This resulted in tons of different forms. I was wondering if they could be bred with Oreo crumbles to make a koi. Well, if you cross Oreo crumbles with powder oranges, you'll get, this is what I got when I did it. I got wild types, the first generation. Some wild types. There were some others that were not crosses, that, you know, there were some that were just Oreo crumbles and some that were oranges. But when I got those wild types, I separated the wild types out, and those wild types produced various things. They produced Oreo crumbles. They produced oranges, but they also produced some orange creams or kois or whatever you want to call them. And I separated those out, and I'm still working on it. But it's working, and it's working very well. So I now have a population of those almost ready to start selling. And thank you, Mark, and thank you for joining us. Newberger, welcome back. So pink duckies, ball python mom. I haven't actually worked with pink duckies at all. Is pink ducky, there's a species called pink ducky, right? Or not the, or are you talking about the, like the version of rubber duckies that are just, have a pinkish color? So that's the thing, you have the blonde duckies and then you have the pink ones. And I think they're, I think that's how that works. So, let's see, Newberger, do you have any tarantulas? I don't, I'm not allowed to. My wife has asked me not to keep tarantulas. She asked me very early on in our relationship. I don't even know if we were married yet. And she said, I asked her what we could keep. I would be keeping pets. And I said, what are things that you can't do? They're deal breaker kind of creatures, no goes. And she said tarantulas and roaches and anything with medically significant venom. So Frosty Inverts, congrats on that Triceratop beetles, and glad it's doing well for you, and glad you're learning a lot about it. Oh, Theropod Hunter, slow worm, so cool. I hope you're able to get a European legless lizard, one of the other species that are okay. And Waver in Darkness, still taking some questions, yep. McGee, do you think Acellus aquaticus would do well in a ghost shrimp neocaridina aquarium? Very possibly. The only thing is I'm wondering if they would outcompete each other, if you'd have some competition, because they tend, you know, they're detritus eaters. All of those, ghost shrimp, neocaridina, and Acellus aquaticus are all doing a lot of the same things. Their niches overlap to some degree. I'm sure they're not completely the same, but enough so that you might get some competition. Might not be too bad, but could happen. Kind of like scuds. If you put scuds in with shrimp, it's not necessarily a great thing. One of them usually outcompetes the other. I kind of think it would be the same with the Acellus aquaticus. Oh, I see what you're talking about, Ruben, where you have 3 different colors on the isopod. I think what happens with Porcellonides pruinosus in most cases, and I've seen some random things in there. I've seen some that I would call maybe a calico. So if you worked with it enough, you might get something. But the idea is that the orange gene, the gene that causes orange, powder orange, cancels out all gray pigment. And then the pied gene cancels out any pigment, whether it's orange or the gray. And so when you put them together and you have both traits active, you just get a pied orange. So the beetle guy doing a large isopod enclosure, zoom ed, low profile tank. What type of isopod do you think I should put in it? I would either put, I'd probably put a larger armadillidium in there, one of the ones that does well with a decent amount of dryness, because you're going to have a lot of ventilation in there. Or maybe one of the Mediterraneans that does well with a lot of ventilation. You could do Magnificus in there maybe. So Waver in Darkness, I would worry a little bit about bat guano, not so much for the isopods. I think that's not necessarily, well, there is a possibility that you could have ammonia, too much ammonia production, depends on how much. Just putting straight bat guano in there would be more dangerous, obviously. But I worry about zoonoses, like illnesses you could get from bat guano, honestly. I don't know, if it were composted, I'd feel a little better about it. And thank you, Fishroom Fever, for getting that like button. It looks like we've got 38 people who found it. And Just Bugs, yes, thank you, almost 57K. I'm hoping within a week or so I will hit 57,000 subscribers. I'm pretty excited. So Nolan Hutt, do you think you could keep mealworms and millipedes together? Well, in certain conditions, yes. In a big enough enclosure with moisture gradient, yes. Would it be ideal? Possibly not. Could there be potential problems? Yes. Mealworms tend to like it a bit drier than millipedes do in general, but with a big enough gradient, you might get it to work. Okay, so Polythanmon was referring to the species known as pink ducky. Then that is one I don't know about. I have not kept that one. Okay. I think you keep any norms? Norms and millipedes together? Nolan, I think autocorrect attacked you. So what did you mean by norms? And we'll see what we could come up with. So Miles Fitzgerald getting any not frequently mentioned beginner tips. Not frequently mentioned beginner tips. I try to mention all the tips I can think of for beginners really frequently. But one thing that I think doesn't get mentioned outside of what I have talked about in my video is that your isopods are not necessarily going to be very visible and don't need to eat a whole lot in the beginning. And so people get upset. Beginners are like, they're not eating anything and I don't see them. And I just remind them to be patient. Because that will change with time as the colony grows. They'll be eating a lot more and you'll be seeing a lot more of them. So Just Bugs. Reptile, well, I think bioactives really help with any odors that you might be dealing with. So Newburger. It depends, because careless seller can mean a lot of different things. And isopods are fairly resilient. So I think sometimes you can get a colony that's not necessarily thriving when you get it, and you can nurse it back to health for another days, and then it will be amazing. So that happens. And Mr. Zissou, there you go. For ball python mom, I think this is the problem, is that Pak Chong can sometimes be called pink duckies, and blonde duckies are sometimes called pink duckies. It's that name train wreck I was referring to before. So yeah, and I regrettably don't keep Pak Chong, or I don't keep Pak Chong. I do keep duckies, which if they're the same species, then I do keep them. I don't know if I have any great tips for them, but I'll try to think of something. If you have any specific questions about that, we'll see. So what do you recommend as the main water source and food source for desert beetles? So for my desert beetles, I do moisten a corner of the enclosure every week, and I think they get some moisture from that. I also use bug jelly. I get bug jelly from Bugs in Cyberspace, and I offer that. And I also use carrots and or organic carrots and or sweet potatoes chopped up and added. That's water source and a food source. Those are all water sources and food sources. And then I give them dog food, and sometimes I soak the dog food. So again, a hydration source as well as a food source. It's a main food source. I also sometimes give dead crickets, and they really like that. And dead mealworms and dried shrimp as food sources as well. And various other things, fish food pellets sometimes and stuff like that. So ball python mon, what happens when you cross dairy cows with Porcelia laevis white? My understanding is that Porcelia laevis white was isolated from low expression dairy cows, meaning they were expressing low quantities of the black, and there's something genetic going on there. And that I occasionally get the low expression Porcelia laevis in my dairy cow enclosure, And so if you were to mix them, you'd probably just get, it wouldn't be a dramatic thing going on, because you'd probably get a dairy cow population, pretty normal expression, except for just a higher incidence of the white ones in that colony. I think that's what you'd end up getting. So Chenhua 1, do you need to clean up isopod enclosure? You don't need to clean it in the sense that you don't need to remove the frass, like spot clean it like you would for a reptile enclosure or something like that, but you do need to periodically replace the substrate, but it's fairly infrequently. Something you might do every 3 months, every 6 months, depending on the situation, or even less frequently. The substrate does eventually get exhausted, and you do need to replace it, and you don't necessarily need to replace it all at once. I usually do half of it at once or so, but hopefully that answers the question. So you do need to change out the substrate periodically, but it's not the same side of cleaning, and you might want to wipe down the container as dirt and dust and things build up on the sides. Sorry. Gotta get a drink. Okay, here we go. Sorry, I'm so behind on the chat. Sorry, I'm missing that. I'm missing bits of the chat. What happens when you mix dairy cows with sheep? Probably would be a bad mood. Okay, the beetle guy. I think it was Peter at Bugs in Cyberspace who mentioned this, that you can keep mealworm beetles like desert beetles, and you can keep them with desert beetles because they do fairly well in a xeric environment, and they can handle the warm temperatures, and they do fine, and they actually are kind of fun to keep that way. So I don't think there's anything wrong with it. If you're keeping them with other species of beetle, you're probably going to encounter, more predation on each other's eggs and larvae and stuff like that. But blue death fanning beetles will also predate their own eggs, and the larvae will predate each other, so it might not be that big of a deal. I don't know. I am sorry. Really got to keep this throat hydrated. I don't know what's going on today. But hopefully that basically gave you the idea. How do you get springtails to breed super fast? Well, I would say feed them every 24 to 48 hours and moisten that food a little bit, and make sure they get plenty of ventilation in that when you open the container, you're doing that often enough that they're getting enough air. I find that if I feed them every 24 to 48 hours without fail, they will just go through the roof in terms of speed. You have to have a good food too, but that seems to help. Yep. And Mr. Zissou, that's a good point, 503, about the mealworm beetles and morio beetles in the desert beetle enclosure. So yeah, I think it's a good idea. You just have to keep an eye on them because they might breed more than some of the others. So Mr. Zissou, I know you do rove beetles with isopods? Yes, I do. But have you ever tried superworm beetles in a pod enclosure? Yes. And the jury is not quite out yet on how successful that is. I will say that I will often use a superworm or a few superworms and throw them into a container when I want to culture good bacteria, because superworms have gut bacteria and they also eat a lot, for their size and whatnot. They're pretty active creatures and eat a lot. So when I want to get a container going, I'll put everything in there except for the isopods. I'll put the springtails in, and I'll put a superworm larva in, sometimes more than one, and then I'll let them do their thing. Live in there, and I will feed them and whatnot. And they're producing frass, and the springtails are feeding on the frass. They will shed their skins if they're young enough, and the springtails will feed on the skins and whatnot. And this is helping to build up a robust microbial population. So ideally, what I will do then is when I'm getting ready, the colony has been going like that for a month or two. Springtails are usually going like crazy at that point. And many times the superworm beetle has pupated in there and then turned into a beetle. That's kind of why I want to put one in there, because I don't want a breeding colony of superworms in there. But a lot of times I'll just put one, so then I can find that beetle, pull it out, say it's ready to put isopods in, and the bacterial population has been going well, so it's not going to be a sterile enclosure, because you don't really want to put isopods straight in a sterile enclosure. So I think it's a good way to make it work. It seems to work pretty well. And Beetle Guy and 503 talking about the Mario beetles. I've noticed that they'll drink water right out of a dish very readily, and I think that helps. I will keep them in with my leopard gecko. It's pretty dry in there. The lower levels of the substrate and the bioactive substrate, which is biodude substrate, are more humid, and they're perfect for the larvae to find a place to pupate and whatnot. But yeah, they will drink the water right out of a dish, and they seem to do well. And McGee, good question about the isopods interbreeding for lack of genetic diversity hurts them. I think there's a lot of factors at play there. Oren McMonagle mentioned that when he is trying to isolate a morph, he has found that there are problems when he tries to remove too many at once, too many of the hets at once, to just isolate the pure ones, that he can find that he ends up with a weak strain. So I think it can happen. So good question. It's hard to say. I haven't encountered any problems that I have noticed that are necessarily deriving from that issue. Some strains of isopods started with very, very small numbers. And I think that's part of the reason why it's good to start out with a larger count, so you have a little bit more genetic diversity rather than like a 6 count. If you start out with a 10, 12, 15, 20 count, you're doing better, I think. That can help prevent that. So lettuce wrap. I think keeping active monitors would be really cool. Right now I don't have a place to put them, but if I had time and place to put it, I would be interested in active monitors. I think they're very cool. So Nolan, do you know why my mealworms are not pupating? That is interesting. Generally, I have not had an issue keeping them from pupating. I have had issues if the thing is too dry or anything like that. It can, or the larvae don't have enough food, they will nibble on the pupae and kill them. And I've had that issue in the past if I'm not careful, but I haven't had issues with them not pupating. From what I understand, there are some mealworms out there available that are sometimes treated with something that prevents them from pupating. Could be that issue, not sure. Thank you for joining us, Miles. So ball python mom, what do you do with your used pod substrate when you replace it? Despite trying to get them all, there always seem to be babies left in it. True enough. So it's generally a good idea to put that into an enclosure, a container of some type, and moisten one little corner of it and put a little hide in that corner, and wait a week or two, continuing to keep that area moist, and then checking out that area, and keep doing that for a number of weeks until you're reasonably sure you got all the babies out. Once you have done that, then you can put it in the freezer, freeze it for 72 hours, and then you can dispose of it. It can go in a garden or something like that without real worry that it's going to cause any issues. Hey, Tarantula Collective, nice to see you here. Got a super chat, thank you so much. I'm excited for a project that involves both of us. I won't reveal too much about it, but thank you for your help with that, Richard. I'm looking forward to the results of that. So great to have you here on the live stream. And thank you for getting back to me so quickly too. I know you're a busy guy. You got back to me fast. So very excited for that. So, and if you guys have seen, how many of you guys have seen the Exotic Pet Collective, Richard's other channel? He's got some very cool stuff. I've been enjoying watching your podcasts or listening, whether, whichever one I happen to be doing at the time. I've been having a lot of fun with those. They're always fun, and you had some really fun ones just come out. So thanks again, Richard, for that. And I really, really appreciate your support, Richard. It's awesome. Makes me happy. Jade X. Do you have any scorpions? I do. I have an Asian forest scorpion and a desert hairy scorpion. Isotantulus. Oh, I need to see that. I'm getting a strange but very intriguing mental image right now of a cross between isopods and tarantulas. That sounds pretty cool. I might be able to keep those. Good. Well, we got a lot of people who do listen and or watch to the Exotic Pet Collective. That's awesome. So Mr. Zissou, I can't actually ship Jestroy right now. So, okay. That's awesome. So Warren watches Richard too. Excellent. Dave's Little Beasties. I'm not sure if I know that one. So beetle guy, why scorpions and not tarantulas? This confuses me. Well, something my wife has said to me is that there were a lot of things she just wasn't really particularly aware of when we got married, and so she didn't know to say, oh, don't get this either. But it's too late now. So Michael Hare, have to blame you for getting me into isopods. Got 2 cultures going now and can foresee more in my future. Excellent. Thank you for the super chat. Much appreciated. And yeah, if you've got 2 already, you know, it's just a matter of time before you get more. What plants have you had the most success using with leopard geckos? So far it's been, there was a Kalanchoe. Is it called Kalanchoe? I'm not sure how it's pronounced. I don't hear it. I just see it spelled. That one went crazy for a long time. And I actually had to remove it because it was growing too crazily in there. That has been the one that was most successful for me. I've had other plants do okay in there for a while. Some aloes did okay in there. I think I'm going to try just normal aloe barbadensis in there and see how that goes. Right now I don't have any plants in there, but I have had some. So ball python mom, you actually have a lot of different types of isopods living together. That's interesting. That can be rare. Website where you could give me some armored delirium. I think autocorrect has struck again. That's what I'm thinking. You know, what kinds of succulents have you had success with in your leopard gecko bioactives? I'm interested. Armadillidium isopods. Oh, maybe armadillidium isopods? I have a website where I sell armadillidium isopods, if that's where we're going. The autocorrect is being naughty today. But yeah, Isopod shop, my Isopod shop is isopodsrus.com, or you can go to aquarimax.com and it's my price list. Either way will get you there. Wow, we're getting close to the end. That's kind of surprising that we got so close to the end so fast. I'm going to check one more time to see if there are any other Patreon submissions. I think we're probably good, but I'm just going to check because I'd hate to miss one. Nope, I think we got them all. That's cool. Okay, so snake plants aren't true succulents, that's true. But I can see them doing well. I have a snake plant in with my garters. I should maybe put it, I have a billion snake plants. Seems like I should put some with I was thinking of doing that actually. Putting a snake plant in with my leopard gecko. One of the shorter, you know, morphs like Hanai or something similar to that. So maybe I should do that. And yes, Jestroy love. It's hard to beat that species. Oh, Heather's here. Excellent. So let me know what your daughter thinks about what you Instagrammed me about. No hurry, obviously, but interested to see how that goes. Yeah, we're just about to end up here. Got a lot to get through. I have a big project that I'm working on tomorrow, and got to go get ready for that. So Let's see. Theropod hunter. That comment. Isopods are S jingle, where a herp can be a herp. So, oh, that's funny. All right, well, I guess the time has come, and I should head out. Oh, Taylor Swift scorpions, yes, indeed. And I actually keep snake plants with New Caledonian geckos. Yes, good point. Snake plants do really well with them. They're very adaptable if they have decent drainage. So, all right, everyone, thank you so much for joining. Really appreciate your presence. Keep an eye open for some fun things coming out. Thank you for those of you who sent the super chat, and just thank you all for watching. And have a great night. Enjoy the rest of your week, and keep an eye peeled for my next video on Friday.